So we are from the National Agricultural Research and Innovation Center, from Agro-Environmental Research Institute. And I would like to start my presentation with a quotation for the European Environmental Agency. It says that spending time in high-quality natural environments fosters good health and well-being. Nature supports life through a broad range of ecosystem services, including provision services such as fresh water, regulating services such as pollination and climate regulation, and cultural services that provide opportunities for recreation and relaxation. However, the quality of natural environments is highly affected by human acts like overpopulation, pollution, deforestation, etc., etc. Environmental pollution is defined as the contamination of physical and biological components of the Earth's or atmosphere system to such an extent that normal environmental processes are adversely affected. We distinguish point and non-point pollutions. Point source pollution is easy to identify. As the name suggests, it comes from a single place. Non-point source pollution is harder to identify and harder to address. It is a pollution that comes from many places at all once. On this slide, you can see major environmental problems, including different aspects of air pollution, like global change, climate change, or urban air pollution, of water pollution, biodiversity, depletion, waste production, and food supply problems. The tool for estimating likelihood or probability of adverse outcome, or even due to pressures or changes in environmental conditions resulting from human activities, is a risk assessment process. Two different types of risk assessments are presented on these slides. The first is ecological risk assessment that aims to protecting organisms, communities, and the ecosystem. It is applied to all man-made chemicals and their degradation products as well, and natural contaminants that are poisonous or can be toxic above certain threshold concentrations. It starts with the problem formulation phase that was first introduced in the context of ecological risk assessment for a very simple reason, to constrain the focus of environmental risk assessment on the key questions. The second step is an analysis phase, when we try to determine the exposure and the ecological effects. And the third main steps is to characterize the risk. Of course, after that, it is important to communicate the results and try to manage the risk detected. The human health risk assessment consists of 4 main points. The first is toxicity identification. The second is dose response assessment and exposure assessment. And of course, the last step in this case, also the risk characterization. Ecological risk assessment of chemicals or agrochemicals are evaluated by standard procedures, as it is done with other chemicals. This approach is similar to the evaluation of risk for humans, except for one important difference is human risk assessment aim at protecting individuals, whereas ecological risk assessments aim at protecting ecological structures, whether population, communities, or the entire ecosystem. When we talk about risk assessment, it is important to clarify the difference between hazard and risk. I think that this simple picture is good to understand the difference. So hazard is just the fact that certain chemicals can be toxic. On this graph, it is represented that a shark in a sea is a hazard. However, when you're swimming with the shark is a risk. So risk is the hazard and exposure together. On this graph, you can see the basic steps again of the risk assessment based on the directive from the European Commission. The risk assessment of chemical compounds includes a lot of steps. First step involves collecting data on the type of hazards such as a possible environmental damage and human health effects. As an attempt to quantify the potential danger posed by chemicals, a variety of toxicity tests have been devised. Some of the recommended tests involve experiments with subsets of natural systems such as microcosmos or with the entire ecosystems. The majority of testing new chemicals for possible effects have been confined to studies in the laboratory on a limited number of test species. Results from these laboratory assays provide very useful information for quantification of the relative toxicity of different chemicals. The second step implies knowledge of not effective concentration and lethal doses or lethal concentration or effective concentration values. Information in this case can be found by a laboratory experiment, or the data can be obtained directly from the scientific databases or via online data searches. It is very difficult to completely know about the chemicals' effect on all levels, from cells to ecosystem, as some effects are associated with very small concentration. For example, the endocrine disruptor effects. So in this point, it is very important to own a good analytical methods with very good sensitivity and limit of detection for determination of these contaminants from the environmental matrices like soil, surface water, and so on. The third step is application of so-called safety factors. It can be between 10 and 1000. If we have good knowledge about the chemical is available, then safety factor of 10 may be applied. If, on the other hand, it is estimated that the available information has a very high uncertainty, then a safety factor of 10,000 may be recommended. In this table, you can see an example that in which case, which assessment of safety factor are recommended. If you have, for example, one short term, it means that an acute test of 3 trophic level, fish, plankton, and algae, because it's a short-term effect, you have to apply 1000 assessment factor. However, if you see the last row, if you have long-term studies from at least 3 species, normally the same fish, daphne, and algae, these are different indicator taxonomy groups for different trophic levels. You are allowed to use assessment factor 10. So the more information you have, the less safety factor is required. The step 4 is give the question that what are the sources and quantities of emission. The answer requires knowledge of the production, the use of considered chemical compounds. including an assessment of how much of the chemical is wasted in the environment by production and use. In step 5, sorry, there is a question that what is the actual exposure concentration? The answer to this question is the predicted environmental concentration. This is the PEC on the graph. This exposure can be assessed by measuring environmental concentration by different instrument analytical methods, or by a model when the emissions are known. The use of models is necessary to most cases, either because we are considering a new chemical or because the assessment of environmental concentration requires a very large number of measurements in the laboratory and to determine the variation of in concentration. The ratio of PEC and PNEC. PNEC is the predicted no effect concentration. So the combination of these two parameters results the so-called risk quotient. It should not be considered an absolute assessment of risk, but rather a relative ranking of tasks. The ratio, if it is lower than 0.1, it is considered that insignificant risk. If it is between 0.1 and 1, we talk about low risk. If it is between 1 and 10, we talk about moderate risk. And if it's higher than 10, we talk about high risk. The environmental hazard of a substance is classified by 3 characteristics. The first is a presence ability to resist degradation in the aquatic environment. The second is a bioaccumulation, bioconcentration factor in adipose tissue of aquatic organisms, and the toxicity potential, the poison aquatic organisms. The step 8 asks the question that what is the relation between risk and benefit? This analysis involves examination of socioeconomic, political, and technical factors which are beyond the scope of this volume. And the last step asks the question that how can the risk be reduced to an acceptable level? To answer to this question requires the technical, economic, and legislative investigation. Assessment of alternatives is often an important aspect in risk reduction. Environmental monitoring describes the process and the activities that need to take place to characterize the monitor the quality of the environment. Normally, or in general monitoring, when it is done in order to gain information about the present levels of harmful or potential harmful pollutants in discharges to the environmental within environment itself and in living organisms as well. Monitoring may be carried out to assess pollution effects on man and his environment, and in order to identify any possible cause and effect relationship between pollutant concentration and health effect. An important issue that stems for the evaluation of risk of agrochemicals is the management option available to mitigate or eliminate the hazards that these products introduce into the environment. Here, I call it the main task that risk management part of the whole risk assessment process responsible for. The most basic management option for any agrichemical is the application rate and frequency of usage of a product. Here I show you on this slide an example that risk management steps or tasks in case of plant protection products considered in integrated pest management. For example, restriction to the type of crops the chemical products could be applied, and limits the frequency of application of a product during a crop season, specific instruction about the mode of application, type of spray nozzles, planters, timing of application to avoid harm to bees, establishment of vegetated barriers or other types of buffer zones to capture spray dip that could fall onto neighboring land and water bodies. And last one is establishment of vegetated ponds and or sediment traps to capture runoff loaded with residues and allow the natural remediation of these. Thank you very much for your attention.